Good evening, everyone. In the last lecture about the biomolecules, we study about the lipids and the proteins. And also we studied about the carbohydrates, the unit of which is monosaccharides. So in today's lecture, we will learn about another biomolecule, which is nucleotides, and further we will learn about nucleic acids. So starting with the introduction of nucleotides, nucleotides have three characteristic components as we discussed in the first lecture also. So the major components of the nucleotides are they contain a nitrogenous base, that is a nitrogen containing base, a pentose sugar and a phosphate molecule. So the molecule without the phosphate group is called a nucleoside. So if from a nitrogenous base and a pentose, if we only have a nitrogenous base and a pentose, it is called as a nucleoside. But when a phosphate group is added to this nitrogenous base and the pentose combination, then the complete entity is known as the unit is known as the nucleotide. The nitrogenous bases are derivatives of the two parent compounds, which are pyrimidine and purines. So out of the four nitrogenous bases that we will discuss further, two of them comes under the category of purine and two of them come under the category of pyrimidine. So firstly, the nucleic acids. So nucleic acids are the polynucleotides. They are the macromolecules that consist of nucleotides and that are formed by the polymerization of the nucleotides. So F. Mischer was the first who discovered the nucleic acid in nucleus and in nucleus of the pus cells, and he named it as nuclein. Then the term nucleic acid was later coined by the Altman. So this was the basic introduction about what are nucleotides and what is a nucleic acid. So as we discussed that the nucleotide has a pentose sugar. So in nucleic acids, which are DNA and RNA, that is deoxyribonucleic acid, and RNA stands for ribonucleic acid. So as the name suggests, the pentose sugars which are present in the RNA and the DNA, which are also the genetic materials. So in the RNA and DNA, pentose sugar molecules are present. And these pentose sugars in the RNA is the beta ribose sugar, while in case of DNA, it is beta 2-deoxyribose. So we can see the differences clearly in the RNA and DNA. So in this, we can clearly see the differences between the sugar molecules present in the RNA and the DNA. So in the RNA, we can see that it is a beta ribose and there is a hydroxyl group which is present at the second carbon position. While if we look into the pentose sugar which is present in the DNA molecule, we can clearly see that it lacks a hydroxyl group at the second carbon position. So therefore, it is named as beta 2-deoxyribose, and therefore, the nucleotide or the nucleic acid is known as the deoxyribonucleic acid. So this was about the pentose sugars, which are the part of the nucleotides that are present in RNA and DNA. Now, next we have nitrogenous bases. So as I discussed that there are all the nitrogenous bases can be classified under the two parent compounds, which are purines and pyrimidines. So purines are double membered ring structures, while pyrimidines are single membered ring structure. Both purines and pyrimidines contain the nitrogen and they are therefore known as nitrogenous bases. And under the purine, the two nitrogenous bases that comes under the purine category are adenine and guanine. So the easy way to remember is GAPU. So G and A, which are the short forms for guanine and adenine, comes under the category of purine, while the remaining ones, that is cytosine, uracil and thymine, comes under the category of pyrimidines. That is, they contain a single ring structure. So all the nitrogenous bases which are present in the DNA or RNA comes under the category of purine or pyrimidine. And we will later see that a purine will form a bond with a pyrimidine molecule in the DNA molecules, which are double-stranded molecules. So the next is the complete unit nucleotide. So as I mentioned, that nucleotide is a combination of a nitrogenous base, a pentose sugar, and a phosphate molecule. So if I talk about here, since the deoxyribose sugar is represented. Therefore, it is a deoxyribonucleotide. So basically in this nucleotide, we can see that if it is, this is the nitrogenous base, and the nitrogen base or the nitrogenous base, which is guanine, adenine, purine, thymine and cytosine. So they may form a bond with the first carbon of the pentose sugar. So we can see that the 9th carbon of this purine ring is forming a bond with the first carbon of the pentose sugar, and this leads to the formation of a nucleoside, which is a combination of a pentose sugar and a nitrogenous base. So nitrogen of the first place N1 forms bond with the sugar in case of, if we talk about the pyrimidines, so this nitrogen at the first position forms a bond, while if I talk about the purine, which is shown in this structure, so the nitrogen at the 9th position forms a bond with the sugar. So it is important to remember that if we talk about the purines, in purines, the nitrogen which is present at the, which is named as N1 or which is present at the first position, forms the bond with the sugar, while in case of pyrimidines, purines, it is the 9th place nitrogen that forms a bond with the carbon 1 of the sugar. Phosphate forms the ester bond with the, which is a covalent bond with the 5th carbon of the sugar molecule to form a complete nucleotide. So in a complete nucleotide, we see that there is a nitrogenous base that may form a bond in case of pyrimidine with the first nitrogen, and in case of purines with the 9th nitrogen. So they can form a bond with the first carbon of the sugar molecule, which is present in the pentose sugar. In case of DNA, it can be a deoxyribose. In case of RNA, it can be a oxyribose, which contains hydroxyl group at the 2nd carbon atom. So after this, if there is a phosphate which is attached to the 5th carbon atom through the ester bonds, which is a covalent bond, then it leads to the formation of a complete nucleotide molecule. So a complete nucleotide is basically a combination of a nitrogenous base, a sugar molecule, and a phosphate molecule. Next, we see that we know that nucleic acids are formed by the formation of a linear chain by the nucleotides. So these nucleotides are linked together through a phosphodiester linkage. So here is the representation of a DNA molecule and the RNA molecule. There is a difference between the nitrogenous bases that are present in the DNA and RNA. So in DNA, except thymine, uracil is, in case of DNA, thymine is present, but in case of RNA, except thymine, uracil, there is a difference in one of the nucleotides that is present in the DNA and RNA. So in DNA, it is the thymine which is present, while in case of the RNA, it is the uracil which is represented by U is present. So in case of DNA and RNA, basically, we can see that these are the chains of the nucleotides. That is why they are also known as polynucleotides. So both DNA and RNA have a 5' end and a 3' end. And these DNA and RNA molecules are like this phosphodiester linkage is formed from the 5' end to the 3' end. So the polymerization happens from the 5' end to the 3' end of the DNA or RNA molecule. Now, as we see that how the phosphodiester linkage is formed, the phosphodiester linkage is formed from the 3' of the first of the above nucleotide molecule forms the link with the 5th carbon atom, thus resulting in the phosphodiester linkage. So the phosphodiester bonds, one of which is here shaded in the dark, links the successive nucleotide units, and the backbone of the alternating pentose and phosphate groups in both types of the nucleic acids is highly polar. Now, how do we name it as a 5' end and how do we name it as a 3' end? So we can see clearly that the 5' end of the DNA lacks a nucleotide, and similarly the 3' end of the basically the 3' end of the DNA also lacks a nucleotide. So when there is a lack of nucleotide at the 5' position, then it is known as the 5' end, and when there is a lack of nucleotide at the 3rd position, then this is known as the 3' end in case of both DNA and RNA. So this was the differences. So in case of DNA and RNA, which are the polynucleotides, successive nucleotides are linked through the phosphodiester linkages. And in case of DNA and RNA, the only difference that lies between the DNA and RNA about the nucleotides is that in case of DNA, thymine is present, while in case of RNA, instead of thymine, uracil is present. Rest, all of the nucleotides are same in both the cases. The other difference is about the pentose sugar. In case of DNA, we can see that it is the deoxyribose sugar which is present, while in case of RNA, it is the ribose sugar which is present. So this is the structure of a deoxyribonucleic acid, or in short, DNA. So we can see that DNA in most of the cases is a double-stranded molecule, and in these double-stranded molecules, there is a base pair formation. So by base pair, we mean that the base pairs, nucleotides which are present on one strand of the DNA, forms the pairing or the hydrogen bonding with the nucleotides that are present on the other strand of the DNA. And they forms a bond in a specific combination only. Like, it's not like that A can form adenine. A, for example, adenine can form bonds with any of the nucleotides. They'll form a bond specifically with the thymine, which is present on the opposite or the complementary strand of the DNA. So in case of deoxyribonucleic acid, or in case of DNA, we can see that the bond formation. So as I mentioned, that the bond is formed between a purine and a pyrimidine. So in this case, we can see that the purine ring is the adenine, while thymine is a pyrimidine molecule, and therefore there is a bond formation between the adenine and thymine molecule. So whenever there is a bond, hydrogen bond formation between the A and T, so there are two possible, there are two positions where the hydrogen bond formation may takes place, and hence A and T are there are two hydrogen bonds which are formed between the adenine and thymine molecules. While if I talk about the cytosine and guanine, which is also present in the nucleotides or the DNA, then cytosine and guanine are linked through 3 hydrogen bonds, as we can see clearly in this diagram. So therefore, we can say that the adenine forms a base pair with the thymine and cytosine forms a base pair with the guanine. Both of these strands are linked through the hydrogen bonds that are formed between the nucleotides, and this complete unit is known as the base pair. Also, the number of hydrogen bonds between the adenine and thymine are 2, while the number of hydrogen bonds between the cytosine and guanine is 3. So therefore, if there is a more number of cytosine and guanine which is present in a DNA, more the strength of the DNA. Why? Because there will be more number of the hydrogen bonds. So that DNA will be more difficult to break or to tear apart. While if as compared to the one in which the ratio of A and T is more as compared to C and G. So this was about the DNA. And if I talk about the RNA, so RNA is mostly, in most of the cases, the single-stranded molecule in which there is no base pair formation, and instead of thymine, uracil is present. So these are the nucleotides. So these are the nucleotides that are present in the DNA. So these are the symbols of the different nucleotides that are present in the DNA, and they can be represented by A, or DA, that stands for deoxyadenosine, or deoxyadenosine monophosphate. So basically, these are the different names that we can give to this nucleoside. The nucleoside is deoxyadenosine, and the nucleotide is deoxyadenylate, or also known as deoxyadenosine 5' monophosphate. Similarly, we have guanosine. So we have guanine molecule. So the nucleoside is deoxyguanosine, and the nucleotide is deoxyguanylate, or deoxyguanosine 5' monophosphate. The next nucleotide which is present in the DNA is deoxythymidine, or the nucleoside is known as the deoxythymidine, and the nucleotide is known as deoxythymidylate, or deoxythymidine 5' monophosphate. And the next nucleoside which is present in the DNA is cytosine, or deoxycytidine, which is also known as deoxycytidylate or deoxycytidine 5' monophosphate. So basically, these are the nucleotides that are present in the DNA molecules, and we can see that all of them have a pentose sugar, which is a deoxyribose sugar. So all of them have a deoxyribose sugar, and all of these nucleotides are connected through each other, either through the hydrogen bonds if they are present in the complementary strand against each other, or if they are present in the same strand, then through the phosphodiester linkage, they forms a successive link resulting in the polynucleotide, which is the DNA molecule. The next is the nucleotides that are present in the RNA. So these are also known as the ribonucleotides. So similar to DNA, there are 4 ribonucleotides that are present. But as I mentioned, instead of thymine, so instead of deoxythymidine, which is the nucleoside present in the DNA, we have another nucleoside which is present, which is known as uridine. So also instead of deoxy nucleosides, we have simply the ribonucleotides, that is the adenosine, ribonucleosides, that is the adenosine, guanosine, uridine, and cytidine. So we can see that uridine, if we look at the structure of uridine, so uridine is nothing, thymine is nothing, but it is 5-methyl uracil, as the methyl group is present on the 5th carbon atom, 5th position of the uracil molecule. So this is the structure of a uracil. And if I look at the structure of the thymine, so this is similar to uracil, except for the fact that at 5th position, the methyl group is present. Therefore, thymidine is also known as, like this thymine molecule is also known as 5-methyl uracil. So these are the ribonucleotides that are present in the RNA. These are as follows: adenylate, guanylate, uridylate, and cytidylate. So, and RNA in most of the cases is a single-stranded molecule, and therefore, as compared to DNA, it is comparatively more prone to the mutations. And from DNA to RNA, the process of the formation of DNA to RNA is known as the transcription, and this RNA then contains the information about the proteins. So the process of formation of proteins from the RNA is known as the translation. Next is the hydrogen bonding patterns in the base pairs. So this I have also described. So this hydrogen bonding pattern, which I described earlier, also is defined by the Watson and Crick. So this is also known as Watson and Crick base pairing. And we can see that adenine forms the base pair with the thymine, and this adenine and thymine molecules are basically connected to the 2 hydrogen bonds that are formed between the adenine and thymine. Also, we can see that cytosine forms a bond with the guanine, and these cytosine and guanine molecules are connected through 3 hydrogen bonds, and therefore providing more strength to the DNA molecule. So this is the hydrogen bonding pattern. And if we look at the basically the central cavity of the DNA, we look into this. So the distance between one strand to the other strand is constant, and it is because always a purine ring is forming a base pair with a pyrimidine ring. So the distance is almost constant. Now there are different forms of DNA which are present. Basically, whatever we studied, we mostly study with regard to the B form of the DNA, which is the most common form of the DNA. But there are different other forms of the DNA that are also present beside B form of the DNA, and they differ in many properties. For example, if I talk about the helical sense, so there are different forms like A form, B form, and Z form. So both A form and B form are right-handed helical structures, while we can clearly see that the Z form is a left-handed helical structure. If I talk about the diameter of these, we can see that A form is the one which is the broadest diameter, so it is around 26 angstrom, while in case of B form of DNA, it is nearly about 20 angstrom, and this diameter is constant, as I mentioned, in the complete DNA molecule. And if I talk about the Z form of the DNA, it is around 18 angstrom. now the base pairs per helical turn differs. So this is this is known as the one turn, and in one turn there are different number of base pairs that are present in the A form, B form, and Z form. So in A form, the base pairs per helical turn are 11, and in B form they are 10.5, or we can say approximately 10. And in case of Z form, it is the 12th number of base pairs that are present in one helical turn. Now, helix rise per base pair is 2.6 angstrom in A form of the DNA. Helix rise per base pair is 3.4 angstrom per base pair DNA. And in Z form of DNA, it is 3.7 angstrom per base pair. So we can say, for example, if I want to talk about the B form of the DNA, since the helix rise per base pair is 3.4 angstrom and per helical turn, approximately 10 base pairs are present. Therefore, the length of one helix is approximately 3.4 into 10, that is 34 angstrom. So it may be asked in the exams as well that what is the length per helix. If it is asked, then it will be 3.4 into 10, approximately 34 angstrom. And if it is asked helix rise per base pair, then the answer will be 3.4 angstrom. One of the other commonly asked questions is that which form of the DNA is left-handed? So it is the Z form of the DNA which is left-handed. Now, the next point for the differentiation is base tilt normal to the helix axis. So how much is the, what is the angle of at which the nitrogen base tilt normal to the helix axis which is present? So it is 20 degree in case of A form, it is 6 degree in case of B form, and it is 7 degree in case of Z form. If I talk about the sugar pucker formation, it may be endo at some positions, or it may be endo for purines, or it may be endo for pyrimidines in different position in case of different DNAs. So if I talk about the A form of DNA, then it is C3 prime endo. If I talk about the B form, then it is C2 prime endo. And if I talk about Z form, then in case of pyrimidines which are present in the Z form of DNA, it is this C2 prime endo pucker formation, conformation of the sugar. And if I talk about the purines, it is C3 prime endo for the purines. The glycosyl bond formation in different forms of DNA. So in case of A form of DNA, it is the anti. In case of B form, again it is anti-glycosyl bond formation, while in case of Z form, so anti, if the pyrimidines are present, then it is anti, and if purines are present, then these are same glycosyl bond formation. So this was the basics of some different forms of the DNA. The most common points that will be asked at this level will be about the helical sense, the diameter, and the base pairs per helical turn, and the helix rise per base pair. So these are some common differences between the different forms of the DNA. So this was mostly about the nucleotides and the nucleosides that are present. And nucleotide, as I mentioned, like polynucleotides, are the another class of macromolecules that play an immense important functions in the survival of the individual, in the survival of the organisms. So basically, this is the summary about the nucleotides and the nucleic acid nomenclatures. So as we discussed, that the bases can be classified under purines and pyrimidines. Under purines, they can be adenine, guanine, and under pyrimidines, they can be cytosine, thymine, and uracil. So we discussed that adenine, guanine, cytosine, and thymine are present both, adenine, guanine, and cytosine are present both in RNA and DNA, while thymine is present only in case of DNA, and uracil is present only in case of RNA. So this was about the nucleic acids, and what are finally the functions of nucleotides. So they have a variety of roles in the cellular metabolism. They are the energy currency in metabolic transactions, the essential chemical links in response of cells to hormone and other extracellular stimuli, and they are the structural components of an array of enzyme cofactors. For example, we will learn like NAD, so NADP. So these are the various cofactors. So these are basically made up of the nucleotides, that is nucleotide, adenine, diflavonate, etc. And they are they function as a metabolic intermediates also. Similarly, they are the constituent of the nucleic acids. So DNA and RNA, which we talk about, these are this DNA and RNA are the molecular repositories of the genetic information, as we know, and the structure of the information about the structure of every protein or which protein will be encoded in the cell at what point of time is decided by the sequence of the DNA and ultimately by the sequence of the RNA and ultimately by the sequence of the DNA. So therefore, the structure of every protein and therefore every biomolecule and cellular component is a product of information that is programmed into the nucleotide sequence of the cell's nucleic acids. So therefore, it is very important that therefore DNA, the sequence of the RNA and DNA, which is defined by the sequence of the nucleotides that are present in them, defines the structure of every protein or every biomolecule and other cellular components that are essential for the cellular metabolism. Also, these DNA and RNA molecules have the ability to store the genetic information in the form of genes. They have the ability to transmit the genetic information from one generation to the next generation, and therefore they are the fundamental conditions. Therefore, they are fundamentally very important for the life. So this was about the nucleotides and nucleic acids. The next in line is the enzymes. So what are enzymes? Enzymes were firstly discovered by Buchner, and he isolated the enzyme which is known as the zymase from the yeast cells. Next, Pune was the one who coined the term enzyme. J.B. Sumner purified and crystallized urease enzyme for the first time from jack bean plant or the lobia plant, and we can say that almost all of the enzymes are proteins, although there are some exceptions. So when we were studying proteins also, we discussed that most of the proteins also function as enzymes. So almost all of the enzymes are proteins, but there are some nucleic acids that may also behave as enzymes, and these are known as the ribozymes. So during the lecture of protein, when we were studying the proteins, we studied that the protein structure is so there is a hierarchy in the protein structure. So we have a primary structure of a protein, we have a secondary structure of a protein, we have a tertiary structure of a protein, and quaternary structure. Similarly, if we talk about the enzymes, since most of the enzymes are proteins, therefore, like any other protein, they also have a primary, secondary, tertiary, and quaternary structures. As in case of proteins, the primary structure is defined by the sequences of the amino acids that are present in the protein. So basically, enzyme also has a primary structure that is defined by the sequence of the amino acids or the protein that is formed. An enzyme, like any other protein, also has a secondary and a tertiary structure, and these tertiary structures basically forms the backbone. And in this tertiary structures, the backbone of the protein chain folds up. This tertiary structure is very important and play very important role. They have a biological activities. So as we studied during that lecture of protein also, that most of the proteins are active in their tertiary state only, and they are biologically they are able to perform biological functions during the tertiary structure formation after the in their tertiary structure state. So in tertiary structure, basically the backbone of the protein chain, or we can say the enzyme, folds upon itself. It crisscrosses and acquires a conformation that has many crevices and pockets. Out of these crevices and pockets, one such pocket is known as the active site. And what is an active site? Which is the most important site in any enzyme and defines the activity of any enzyme. So an active site of an enzyme is a crevice or a pocket into which the substrate fits. So basically, an enzyme interacts with the substrate and it catalyzes the reaction. So an active site of an enzyme is a pocket where the substrate comes and fits, and thus enzymes through their active site catalyzes the reactions at a high rate. So by catalyzing, I mean that they speed up the reaction, and similar to so therefore enzymes are also known as biocatalysts since they are present in the living organisms. Although we also know about the inorganic catalyst that we study in the chemistry, So there are many catalysts that are present, that are used in the industrial applications that help in the acceleration of the reaction, and enzymes are similar to that, but in some of the aspects they differ from the inorganic catalyst. For example, one of the major differences is that the inorganic catalyst works efficiently at higher temperatures, while if we talk about the enzymes or if we talk about the biocatalyst, they tends to get damaged at higher temperatures, except for the enzymes that are isolated from the organisms who normally live under extremely high temperature conditions. For example, the organism that lives in the hot vents and the sulfur springs. So the enzymes that are isolated from such organisms are stable. For example, we use Taq polymerase, which is one of the enzymes that are frequently used in the molecular biology. So this is isolated from the organism that is able to survive at a higher temperature, and such enzymes are stable and retain their catalytic properties even at the higher temperatures, like up to 80-90 degrees centigrades. And thermal stability is therefore an important quality of such enzymes isolated from the thermophilic organisms. Besides these enzymes, most of the enzymes that are isolated from the organisms that survive at normal temperature and pressure conditions, they are not able to bear the such high temperatures, and they tend to get damaged or denature at such high temperatures. So how these enzymes basically helps in catalyzing a reaction? So what is the working principle of the enzyme? So whenever there is a reaction that has to be happened, so it has to pass through the activation barrier to convert into a product. So if I talk about the reactant A, which is having this free energy, let's say X, so it will form a product which is having a lower free energy, let's say Y, and to convert from the reactant A to the product B, it has to pass through a activation barrier, which is the free energy change that it has to cross, free energy barrier that it has to cross. So an activation barrier representing the transition state. So the molecule which is formed at this state is known as the transition state, and because of the very high free energy, it is unstable and it tends to move rapidly into the product. So an activation barrier here represented by the delta G uncatalyzed has to be passed or must be overcome by the reactants in order to convert into the products. So even though the products are stable than the reactants, there is a large negative free energy change that is to be brought. And the energy required to overcome this activation barrier is known as the activation energy. So basically what enzymes do is, enzymes lower this activation barrier and thereby lowers the activation energy which is required to cross the barrier. So we can see that the blue line here represents the conversion of a reactant to product in the uncatalyzed form when there is no enzyme is present, while the red line is the one in which there is a conversion of reactants to products and through the catalytic mechanism. So we can see that there is a decrease in the activation barrier that it has to cross. Therefore, it can cross the activation barrier even at even with the very small change in the energy. So they bind the transition state intermediates tightly, and the binding energy of this interaction effectively reduces the activation energy from uncatalyzed to the G catalyzed. So due to these differences in the activation energy barrier that is created by the catalytic reaction or that is created by the enzymes, so there is a rapid conversion of the reactants to the products, and this is how the enzymes or a catalyst work. So these enzymes basically work by decreasing the activation energy that is required to convert a reactant into a product. So we can see clearly that there is a difference. So the delta, therefore the G uncatalyzed, which is the activation energy which is required in an uncatalyzed reaction, is lowered in the presence of the enzyme, and therefore the reaction proceeds faster. So how the enzyme function? What is the nature of the enzyme action? The catalytic cycle of an enzyme action can be described in the following 4 steps. So this is the diagrammatic representation. So in the first figure, we can clearly see that this gray represents the enzyme, and this crevices or the pockets represents the active site. So active site is the site where the substrate comes and fits and it binds with the enzyme. So basically, in the first step, firstly the substrate binds to the active site, fitting into the active site. So there is an interaction between the enzyme and the substrate. This interaction or the binding of the substrate induces a little bit change in the shape. As we can see that there is a little change in the shape that allows the tight fitting of the substrate into the enzyme more tightly, leading to the formation of the enzyme substrate complex. Now this the active site of the enzyme, now in the close proximity of the substrate, breaks the chemical bond of the substrate and the new enzyme product complex is formed. Basically, by breaking down the bonds of the substrate, it is leading the production of this product, and now there is a formation of the enzyme product complex. And at the last stage, what happens is the enzyme releases the products of the reaction and the free enzyme is ready to reuse, or it can bind to another molecule of the substrate. So one of the important properties of the enzyme or any catalyst is that they are not used up in the reaction. Like they are always, like once the cycle is complete, we can reuse the catalyst or the enzyme again for the another molecule of the substrate. So this is how the enzyme functions. Firstly, it binds with the substrate, and when it interacts with the substrate, there is a change in the shape of the enzyme. There is a little bit change in the shape of the enzyme that allows the substrate to bind in the more tighter way with the enzyme, forming the enzyme substrate complex. Now, since the enzyme and the substrate are in close proximity, enzyme can catalyze the reaction, leading to the breakage of the bonds between the substrate and formation of the product, resulting in the formation of enzyme product complex. Now, this enzyme releases the products of the reaction, and the free enzyme is ready to reuse for the next molecule or the another molecule in the reaction, another molecule of the substrate in the reaction. These enzymes are like there are thousands of enzymes that has been discovered, but they are mostly divided into the 6 classes. Each of these 6 classes have 4 to 13 subclasses, and therefore, if we look into the nomenclature of any enzyme, they will be represented by a 4-digit number, with the first digit representing the class of the enzyme. So these 6 classes or the classes of the enzymes can broadly be classified into these 6 classes and be broadly classified under the category of these 6 classes. The first one is the oxidoreductase or dehydrogenases. So like we will see that whenever there is an enzyme, there will be mostly the suffix SES, that is ases, is used. So oxidoreductases or dehydrogenases are the enzymes that catalyze the oxidoreduction between the two substrates. So if there is a substrate that is in the reduced form and there is a S prime substrate that is in the oxidized form, so it catalyzes the oxidoreduction. So basically it leads to the oxidation reduction reaction they are involved in the oxidation reduction reaction in carrying out the oxidation and reduction reaction. The next is the transferases. So as the name suggests, transferases, that is, these enzymes help in the transfer of a group from one substrate to another substrate. But that group should not be hydrogen, because in that case, the enzyme will come under the category of oxidoreductases or dehydrogenases. So if except hydrogen, there is any other group that gets transferred from one substrate to another substrate, so this will be carried out with the help of the enzymes which are known as transferases. Next is hydrolases. So as the name suggests, these enzymes help in catalyzing the hydrolysis reaction of ester, ether, peptide, glycosidic, carbon-carbon, hydrogen bonds, etc. Next class of enzymes is lyases. So these lyases are the ones that catalyze the removal of the groups from the substrates by the mechanism other than hydrolysis, leaving the double bonds. So Basically, in case of hydrolysis, we will see that there will be the removal of water molecule. But in case like there will be the use of water molecule to break the bond. But if it, like by the any other mechanism, there is a removal of the groups, if, as we can see in this figure, and there is a formation of a double bond as a result, then this is carried out by the enzymes, which is known as lyases. The next is isomerases. So, as the name suggests, it includes all the enzyme that catalyzes the interconversion of optical, geometric, or positional isomers. So, the last class of the enzymes is the ligases. So, ligases are the one that helps in tying up the bond. So, basically, that ligases contains the enzymes that catalyzes the linking together of two compounds. Examples, the enzymes that catalyzes the joining of CO, CS, CN, PO, etc. like different bonds. So basically ligases helps in joining of different bonds, different atoms through the bonds. So these are this is the table that shows the classification of the different enzymes under the 6 different classes, which were oxidoreductases, transferases, hydrolases. As we can see, in the presence of water, the bond is breaking between, like the bond broke between the A and B, and 2 different molecules are formed. Lyases, which is the non-hydrolytic addition or removal of groups. Isomerases, that helps in the intramolecular rearrangement, and therefore the isomerization changes happen within the molecule. And ligases are the ones that helps in joining the two molecules, or the that helps in the synthesis of different bonds. These are the examples of the different enzymes that comes under the category of these different classes. For example, dehydrogenase or the oxidase is the one that comes under the class oxidoreductases. Transaminase and kinase are the ones that comes under the class transferases. Lipase, amylase, peptidase are the hydrolysis enzymes. And decarboxylase is the one which is a lyase. Isomerase or mutase are isomerases. And synthetase is a ligase that helps in the ligation of molecules. Now, enzymes as a whole can be enzymes can be present. Enzymes may be present as a completely protein part, or they may contain some non-protein part as well. So enzymes can be the protein part in such cases where the enzyme has a protein as well as a non-protein part. So in such cases, the protein part of the enzyme is known as the apoenzyme, while the non-protein part of the enzyme is known as the cofactor. So the holoenzyme, basically apoenzyme plus cofactor, results in the holoenzyme or the complete enzyme, which contains both protein and non-protein part. The cofactor can further be divided into different categories depending on what is the nature of the non-protein part. So they can be classified as prosthetic groups, coenzymes, or the metallines. So prosthetic groups are the organic compounds, and they differ from other cofactors in the fact that they are tightly bound to the apoenzyme, as compared to if I talk about the coenzyme or the metallines. So they are the ones that tightly bound to the apoenzymes. For example, in case of peroxidases and peroxidase and catalase, that catalyze the breakdown of hydrogen peroxide into the water and oxygen. Here, the next example is the heme, which is the prosthetic group, and it is a part of the active site of the enzyme, and it is tightly bound to the apoenzyme or the protein part of the enzyme. The next class is the coenzyme, which is also an organic compound, but they form a transient association with the apoenzyme. So this is the basic difference between the prosthetic group and the coenzyme. Both of them are organic compounds, but prosthetic groups are the one that are tightly bound to the apoenzyme, while the coenzymes are the cofactors that bound transiently with the apoenzyme. So coenzymes are the essential chemical components of many coenzymes are vitamins. Example, if I talk about the coenzyme nicotinamide adenine dinucleotide, that is NAD and NADP, they both contain the vitamin niacin. The next class of the category is the metallines. So they contain inorganic metals, metallines, as a part of as a cofactor, and they form the coordination bonds. And for example, if I talk about zinc, zinc is a cofactor for the proteolytic enzyme if carboxypeptidase. So there are different metal ions that may act as a cofactor for different enzymes. And the catalytic activity of the enzyme is lost if the cofactor is removed from the enzyme, and therefore it proves the importance or the crucial role of the cofactors in defining the enzymatic activity. So this suggests that besides the protein part or the apoenzyme, the presence of the cofactor or the association of the cofactor with the enzyme, with the apoenzyme part or the protein part is equally important. Without the presence of the cofactor, enzyme will not be in the functional state. For example, if I remove zinc, if I do not provide zinc in the presence of carboxypeptidase, the enzyme will not be able to function. So basically, apoenzyme, which is a protein portion which is inactive, while the non-protein portion, which is a coenzyme, which acts as an activator. So this protein portion, when interacts with the non-protein portion of the enzyme, it results in the holoenzyme, which is the whole enzyme, so which is in the active form. And the substrate then interacts with the holoenzyme, and this holoenzyme catalyzes the reaction. Now, as I mentioned, that the enzymatic activity is very dependent on different factors, like temperature plays a crucial role, pH plays a crucial role in defining the activity of enzymes. So the factors that affects the enzymatic activity are temperature, pH, concentration of the substrate that is present. So enzyme generally functions in a narrow range of temperature and pH. Specifically, if I talk about the pH, so the range is quite narrow. Each enzyme shows its highest activity at a particular temperature and a pH, which is known as the optimum temperature or the optimum pH for the enzyme function. And both below and above the optimum value, the activity of the enzyme decreases. Lower temperature preserves the enzyme in a temporarily inactive state, whereas higher temperature destroys the enzymatic activity because of the denaturation of the protein by the heat. So basically, the take-home message from this slide is that the enzyme functions at the enzymatic activity of any enzyme is highest at a particular temperature and pH, which is known as the optimum temperature and optimum pH value for that enzyme. The next, for example, if we look into this figure, the enzymatic activity, if we look into the enzymatic activity in comparison to the change in the pH or change in the temperature, so they are both below and above the optimum pH value, the enzymatic activity decrease. Similarly, both below and above the optimum temperature value, which is the value at which the enzymatic activity is highest, the enzymatic activity decreases irrespective of the value. The next factor is the concentration of the substrate. So with the increase in the substrate concentration, the velocity of the enzymatic reaction first rises and ultimately reaches to the maximum velocity. This is the velocity, or this is the time point where all the enzymes are saturated, like there is no active site which is available irrespective of the concentration of the substrate. So therefore, above this Vmax, the velocity of the enzymatic reaction cannot be exceeded, and despite of any further increase in the concentration of the substrate. And after the saturation of these molecules, there are no free enzyme molecules that are present in addition, to bind to the additional substrate molecules. And therefore, once the enzyme reaches this Vmax value, the value becomes constant. Enzymes can be inhibited in various manners, so which is known as the enzyme inhibition. So this can be reversible or irreversible inhibition. As the name suggests, reversible inhibition refers to the temporary loss of the enzymatic activity, which is caused by the binding of inhibitors to the enzyme proteins in a non-covalent manner. And since it is reversible, it can be removed by physical methods, like if I perform dialysis, so the inhibitor can be removed and partially or complete restoration of the enzymatic activity may take place. Reversible inhibition includes competitive, uncompetitive, and non-competitive inhibition. While irreversible inhibition means that the inhibitor covalent, in case of irreversible inhibition, there is the permanent loss of the enzymatic activity, and this is due to the covalent bond which is formed between the inhibitor and the active center or the active site of the enzyme, thus inhibiting the activity of the enzyme. So, irreversible inhibitors cannot be removed by any physical method. Like for example, if we perform dialysis, uh, and there is a covalent bond formation between the inhibitor and the active site of the enzyme, the activity cannot be restored at any cost. So, if I talk about the competitive, uncompetitive, and non-competitive inhibitions in little more details, we can understand them from this diagram, from these diagrams. So, competitive inhibition involves the inhibitors that are analogs or that are similar to the substrate. So, they can compete with the substrate and bind with the enzyme, bind to the active site of the enzyme, forming in the enzyme inhibitor complex. But the name competitive itself suggests that by increasing the competition between the substrate and the inhibitor, we can restore the enzymatic activity. So if I increases the substrate concentration to such a level that most of the enzymes bind to the substrate, it will result in the restoration of the activity and ultimately the product will be formed. Now, what is non-competitive inhibition? So, enzymes, in case of non-competitive inhibition, enzyme can bind to both substrate as well as to the inhibitors. And there is no competition between the substrate and non-competitive inhibitor. After the enzyme binds to the non-competitive inhibitor, it can also bind to the substrate, therefore resulting in enzyme-substrate inhibitor complex. However, if enzyme substrate inhibitor complex is formed, product will not be formed. But if the enzyme only bounds with the substrate, then the product will be formed. So therefore, it reduces the enzymatic activity, but do not completely inhibit the activity. The next is the uncompetitive inhibition, when there is no competition and the inhibitor binds only to the substrate enzyme complex. So once there is a formation of the substrate enzyme complex, the inhibitor can only bind to this substrate enzyme complex. The inhibitor cannot bind to the enzyme only, and therefore further reducing the activity of the enzyme by forming the enzyme substrate intermediate complex that does not result into the product. While the ones that forms the enzyme substrate, only that unit will be converted into the product, therefore reducing the activity of the enzymes. So this was all about the enzymes, and these were the different competitions, inhibition that happens at different levels of the during different times, and depends upon the whether there is a reversible inhibition or there is a irreversible inhibition. Reversible inhibition can further be competitive, non-competitive, or uncompetitive. And today we talk about the nucleotides. So let's have some practice questions. So the first one is nucleic acids result from the polymerization of. So we know that nucleic acids are also known as polynucleotides, and they are the results of the polymerization of the nucleotides. Nucleotides are itself the combination of nitrogen bases, nucleosides, and phosphates. So nucleic acid results from the polymerization of the two nucleotides. Next question is, which of the following is a nitrogen base which is not normally found in DNA? So we know that in case of DNA, the nitrogen bases that represent in DNA are guanine, cytosine, adenine, and thymine. But uracil is the one which is not normally found in DNA. So the correct answer is option number 2, uracil. The next question is components that are common in DNA and RNA are. So we know that both DNA and RNA has a pentose sugar. Both DNA and RNA have phosphate group. Both DNA and RNA have nitrogenous bases, but the nitrogen bases like thymine differ in DNA and RNA. So the correct answer that the components that are sugar in DNA and RNA is the phosphate groups. Which of the following base pairing is wrong? So we know that if we go through the Watson-Crick base pairing method, so A forms a bond with T and C forms a bond with G. The rest, like A may also form a bond with U in case of RNA. So the incorrect base pairing is represented by the A forming a bond with G. Sorry, the correct answer should be A forming a bond with G. The next is nucleotide is a combination of, so yes, nucleotide is a combination of nitrogenous base, pentose and basically nitrogenous base and pentose sugar. So, and a phosphoric acid. So the correct answer is option number 1. The next is, which one of the following statement is correct for inorganic catalyst? We know that inorganic catalysts do not occur in living cells. They are not specific for any reaction is also incorrect. So basically inorganic, they are specific for any reaction. They do not get damaged at high temperature. So this is incorrect for inorganic catalyst. Enzyme catalyze biochemical reactions by, we know that by lowering the activation energy, the enzyme catalyzes the biochemical reaction. So an organic substance that bound to an enzyme and is essential for its activity is known as the coenzyme. Which enzyme is not a protein? So we know that except ribozyme, all of the enzymes are most of the enzymes are proteins. Enzyme inhibition caused by a substrate analog. So substrate analog leads to the competitive inhibition. So with this, I would like to end today's session, and I would like to thank you all for attending this biomolecule series. And with this, we wrap up with the chapter biomolecules. Thank you.